Hi everyone, welcome to the session. This is the fifth virology lecture. Today I'm going to talk about genome expression and genome replication in class 6 and class 7 viruses. And when I'm saying class 6 and class 7, I'm talking about the Baltimore classifications. You remember from the Baltimore classification system that we've studied, when we're talking about class 6 and class 7, we're talking about viruses that reverse transcribe. So they, when expressing and replicating their genome, they engage in a quite unusual genomic process called reverse transcription. That's why they're classified differently to the other viruses on the Baltimore classification system, and that's why we're going to consider them separately. The general principles of genome expression and replications still apply, so the fundamental principles are there. It's just the way these viruses express and replicate their genomes. There's differences in terms of the specifics of what they do. So the general principles in the sense that genome expression involves the production of RNA, and then this RNA is translated into protein, and genome replication involves producing exact copies of the genome with the exception of mutations. These are all principles you've covered, and these principles still absolutely apply. It's just the process of how these things are done. The process is different in these two groups, and that's why we want to consider these groups separately. We've kind of touched on genome expression a little bit in the retroviruses, and we'll talk about the retroviruses first today. The retroviruses belong to class 6. We brushed over it a little bit just because it's such a complex aspect of genome expression and genome replication. These are complex viruses, and reverse transcription is a complex event, so we really need to give it a bit more time, and we didn't really touch on genome replication at all last week for class 6 and class 7. So again, that's what we're going to do today. So we'll talk about class 6 first, and class 6 are the retroviruses, or includes the retroviruses, I should say. As you can see on the Baltimore classification system, the genome is single-stranded RNA, and then we have RT on the end, and this RT stands for reverse transcription. So that means these viruses are different from the other single-stranded RNA viruses because they reverse transcribe, because they engage in this very unusual genomic process. You'll also see some intermediates here. You'll see there's a DNA RNA intermediate, and there's a double-stranded DNA intermediate. We're going to explain that today. We did explain it a little bit the other week because, as I said, we did talk a little bit about retroviral genome expression. If you remember this Baltimore classification system, you remember it's really based upon partly the genome composition, but really based upon how mRNA is produced, so how these genomes are expressed. That's why in each class there's this arrow pointing towards mRNA in this representation of the system. The intermediates are also listed here, and that's what I kind of just pointed out in between single-stranded RNA RT for group 6 and the mRNA that is produced. We have DNA RNA, and then we have double-stranded DNA. So these are the intermediates that are involved in the process of genome expression for these group 6 viruses. So just a little bit of context around these group 6 viruses, because this is really, really important. The way these viruses express and replicate their genome has clear implications for infection. So it has clear implications for the type of infections they cause. You're probably aware that HIV is a virus that infects humans, and in doing so, it causes AIDS. It's a treatable infection, but it's not a curable infection. So it's a chronic, incurable infection that can be controlled with a drug regime. If it's controlled with a drug regime, generally an individual who's infected can live a normal, healthy life. There's no real impact on longevity. However, if it's not controlled, we'll see progression towards AIDS. So HIV is the virus that causes AIDS, and AIDS is the long-term consequence of HIV infection. Not all viruses are chronic and incurable. A large number of human viruses are cleared by the immune system, even the ones that cause pathology, so even the ones that cause disease. A lot of the viruses that can't be cleared by the immune system can be cleared with the help of drugs, so with therapeutic interventions. So why is HIV chronic? Why is HIV incurable? This is one of the things we're going to talk about today, because the reason HIV is incurable is a consequence of the way the virus expresses and replicates its genome. So what we have here is a diagram of the viral replication cycle, essentially, and this is what's happening when the virus infects immune cells in an individual. We can see the virus particle is entering into the cell, then it's producing copies of itself, then these copies are exiting the cell. This process results potentially in lysis of the cell, not always, but it can cause lysis. It can also cause widespread immune changes, and these immune changes combined with the direct lysis result in a drop in immune cell levels in this kind of circulating levels, the number of immune cells in the blood over a long period of time. And again, HIV is a chronic infection, so it does, infection does occur for a long period of time. You eventually see a drop in the number of immune cells when an individual loses their immune cells. So when an individual experiences this drop in immune cell numbers, they become immunodeficient. Their immune system is depleted. They don't have sufficient numbers of immune cells to fulfill immune functions. So they're immunodeficient. They're susceptible to additional infections. And in that state in which they are immunodeficient and they have an increased susceptibility to other infections, we say they have acquired immunodeficiency syndrome, which we abbreviate to AIDS. And again, just emphasizing the point that HIV is a chronic, incurable infection. So it's not cleared by the immune response, and it's not cleared effectively by normal therapeutic interventions. You may be aware of the recent cases of the Berlin patient and the London patient, and these are a couple of cases in which HIV has been cured. These are absolutely valid. They're pretty amazing, and when we go through this lecture and we understand how the HIV as a virus, as a retrovirus, expresses and replicates its genome, we'll understand why the specific medical intervention used in these two cases worked, and why, generally speaking, for most people, HIV isn't curable. There just isn't a therapeutic option available to cure HIV in those people. So I've said there's a link between the way HIV replicates and expresses its genome and the kind of characteristic of infection. So by that I mean one of the characteristics of infection is this chronicity, it's this incurable nature, and we can directly link that to the way HIV as a retrovirus expresses and replicates its genome. And the key is a process called integration. HIV integrates its genome into the host cell genome, and this is really crucial, because what it means is even if we're treating HIV infection with antiviral drugs called antivirals, we're not able to get rid of the integrated HIV genome from the host cell genome. So what antiviral drugs tend to do is they target the replication cycle. So viruses, when they're infecting a cell, they undergo the replication cycle. They make copies of themselves, right? Drugs intervene on that. So drugs target that replication cycle. If we have an integrated HIV genome, then it doesn't matter if you're targeting the replication cycle. It doesn't matter if you're inhibiting replication completely, because the HIV genome is still going to be there. It's still integrated in the immune cell genomes. And as soon as an individual stops taking antiviral drugs, then the HIV genome is potentially going to be expressed again, and you're going to get more HIV produced almost straight away. So it's pretty clear how and why using drugs we can control HIV infection. We can stop HIV from expressing its genome because we use the drugs that inhibit the replication cycle. However, using those drugs, we can't actually cure infection because we can't get HIV out of the genome using these drugs. Again, all these drugs are doing is inhibiting the replication cycle, so it's inhibiting the virus from making copies of itself. They're not getting rid of the viral genome from the host cell genome. Okay, so integration is key. Integration and reverse transcription go hand in hand. So we've said how one of the defining features of these retroviruses is they reverse transcribe. Reverse transcription is a huge part of their genome expression and replication. It's why they're classed in Baltimore classification 6. It's because they reverse transcribe. Reverse transcription and integration completely linked, and it's integration that is essentially the cause of HIV's chronicity. So the cause of HIV being a long-term incurable infection. So what we want to do now is look at these two processes, reverse transcription and integration, see how they link together and see exactly what's going on. What we'll see is these two processes are absolutely integral to genome expression and genome replication in these retroviruses. So they're both tightly linked because they're both central processes in genome expression and genome replication. So first, reverse transcription. And I know we did touch on this the other week, but it's such an unusual and important process. We just need to establish these definitions again, really. So we have the host cell genome at the top here, which, as we know, is double-stranded DNA with a positive strand and a negative strand. Then we have the HIV genome. As we also know, HIV has a single-stranded RNA genome, and this genome is positive sense. So how can integration occur? So I've told you that the reason for HIV's chronicity, the reason that it's a long-term incurable infection, is because the HIV genome integrates into the host cell genome. How can this be possible? Because the HIV genome is RNA and it's single-stranded, and the host cell genome is DNA and it's double-stranded. So how can a single strand of RNA integrate into a double strand of DNA? As it is, looking at RNA and DNA, we know that's impossible. We know that you can't just slip a piece of RNA into DNA. It doesn't make sense. Enzymes won't be able to recognize it. It'll just be a mess. It'll never happen. However, what can happen is we can turn the single strand of RNA into a double strand of DNA using the process called reverse transcription. And this is what the virus does. This is what retroviruses have evolved to do. And this is one of the reasons why reverse transcription and integration are fundamentally absolutely linked, because to integrate the HIV genome into the host cell genome, the HIV must reverse transcribe that genome first. It must produce a double-stranded DNA copy of the single strand of RNA. So it must use the single-stranded RNA genome it has as a template to produce double-stranded DNA, and this double-stranded DNA can then be integrated into the host cell genome because it's the same composition, it's the same type of thing, so it can just slip right in. This is why the process we're talking about is so strange, and it's why these viruses need to be considered separately, because we don't see anything like this anywhere else. It's a completely unusual genomic process. So if we think about what we've talked about so far, we've talked about the genomic process of genome expression, which is producing RNA copies of the genome. So we looked at all the different types of viral genomes. We considered how all these different viral genomes produce RNA copies, and these RNA copies are then translated into protein, and we defined this as genome expression. Then we looked at the genomic process called genome replication, and genome replication is where a genome is being copied into an almost identical copy. So an identical copy in the sense that it's of the same composition and it has the same information, with the exception of mutations, of course. And again, we define this process as genome replication. The genome is being replicated. What we're thinking about now is a very, very different process. We're thinking about integration, and we're thinking about reverse transcription. This reverse transcription, again, it seems non-intuitive because we're going from RNA to DNA. It's a really unusual thing to happen. But again, the clue's in the name, it's just reverse transcription. In the host cell, transcription is when you use a DNA template to produce RNA. And this is just the reverse of that. That's literally why it was called reverse transcription. It's because the virus is producing a DNA copy of the RNA template. You'll remember in earlier lectures we talked about these polymerases, and I think I briefly mentioned this specific polymerase, the RNA-dependent DNA polymerase. I think it was an example in some of the of a potential answer in some of the multiple choice questions you did. Now we want to consider it in a little bit more detail, because this is the enzyme that catalyzes reverse transcription. So I said earlier in this lecture, right at the start, that the principles of genome expression and genome replication still apply. The rules are still the same. It's just the details of how the process is done that's different in these group 6 and group 7 viruses. And this should be clear here. This is another example of that. We're talking about an RNA-dependent DNA polymerase. The naming system is exactly the same as we looked at earlier. It's an polymerase, and the way we name it is we name it based upon the template it uses and based upon the type of nucleic acid it produces. So we know this enzyme is using an RNA template to produce DNA. That's what reverse transcription is. So we call the enzyme an RNA-dependent DNA polymerase. Again, it's using an RNA template to produce DNA. The end product of the reaction that this enzyme mediates is therefore DNA, and this DNA can then be integrated into the host cell genome. Again, hopefully it's clear how the process of reverse transcription, which is this process, is inherently linked to the process of integration, because you can't have integration without reverse transcription. And just another diagrammatic representation here. We have normal cellular transcription on the left. So we have a double-stranded DNA genome being transcribed into RNA, catalyzed by an RNA polymerase, specifically a DNA-dependent RNA polymerase. Then on the right, we have reverse transcription, which is essentially the reverse of that process. So we have a double-stranded DNA genome being produced from a single-stranded RNA template. And again, this process is catalyzed by an RNA-dependent DNA polymerase, you'll see the term reverse transcriptase used. Reverse transcriptase is a bit more of an informal term to refer to the specific enzyme that's used by HIV. So when we talk about an RNA-dependent DNA polymerase, we could really be talking about any enzyme that uses an RNA template to produce a DNA copy. Not that there's many enzymes that do that, and there's no host cell enzymes. But when we're talking about HIV, we use the term reverse transcriptase to refer to this enzyme. So reverse transcriptase is the specific RNA-dependent DNA polymerase that is utilized by HIV, because remember, HIV isn't the only retrovirus, it's just the one that we care most about, really. So the process of reverse transcription itself is complicated, to say the least. So this is a diagram I made, I made this for my PhD thesis, I think. Obviously it's quite complicated. We don't need to learn the details of this. It's really just to demonstrate that it is a complicated process, and to demonstrate that we're going from single-stranded RNA, which is what we have at the top, to double-stranded DNA, which is what we have at the bottom. What's actually going on here is there's just bits of primers moving around. So when we talk about RNA being turned into double-stranded DNA, we think about the polymerase just progressing along the RNA and producing one copy of DNA, then progressing along that DNA and producing the complementary DNA, perhaps. That's kind of a simple way to think of it in our head. But actually it's a lot more complicated than that. The enzyme jumps around and then it moves here and then it moves. It doesn't really matter though. I think this is an interesting diagram. The main takeaway is that we're going from single-stranded RNA at the top to double-stranded DNA at the bottom. So again, the single-stranded RNA genome of the virus is being used to produce double-stranded DNA, and this double-stranded DNA can then integrate into the host cell genome. Okay, so that's reverse transcription. Hopefully that's, again, nice and clear. If it isn't, I'd encourage you to just flick through the slides again. Now we want to go on to integration. So again, reverse transcription and integration inherently linked. We can't really consider one without the other. So what we're going to do now is consider integration. So our starting point for integration is a double strand of DNA. So reverse transcription has already happened. The process has produced double-stranded DNA from the single-stranded RNA genome of the virus. What happens next is this double-stranded DNA needs to be integrated into the host cell genome, and this is the process of integration. So how does this occur? First, the 3' ends of the double-stranded DNA are essentially processed in such a way that enables them to almost attack the host cell genome, so allows them to insert into the host cell genome and override the bonds that exist in the host cell genome. We know nucleic acid has a 5' end and a 3' end, and we know that in double-stranded genomes the ways are opposite. So 5'3" is one direction on one side, and then 5'3" is the other direction in the other, and that's what we can see outlined here. So in the double strand, which is linear, we have both 3' ends being processed, and then we have integrase mediating the insertion of these ends into the host cell genome, and this insertion event essentially ends up with the entire viral genome being able to slip into the host cell genome, and integration, again, that's really the best word for it, it integrates into the host cell genome. And this process is mediated by an enzyme that we briefly mentioned recently, and that enzyme is integrase. It's important we understand that integrase isn't a polymerase, so there's no nucleic acid being produced here. There's just the insertion of the double-stranded DNA, and this double-stranded DNA was already made during reverse transcription. So at this stage, there was just the insertion of that double-stranded DNA into the host cell genome. So integrase is not a polymerase, it's not an RNA polymerase or a DNA polymerase. It's just a run-of-the-mill enzyme, if you like, that is catalyzing a specific reaction. And the specific reaction that is being catalyzed is the insertion of the viral genome into the host cell genome. So where does this integrase come from? Well, a long time ago, I think in the first lecture, you'll remember I said that some viruses will have functional enzymes packaged into the virus particle. So they will have functional enzymes within the capsid. One of these enzymes is integrase for HIV. So for these retroviruses, including HIV, there is an integrase packaged within the capsid, and there is also an RNA-dependent DNA polymerase, which for HIV is reverse transcriptase. So these two enzymes that are required for reverse transcription and integration are packaged inside the virus capsid. Okay, so that's integration covered. It's not a hugely complicated process itself, and you don't really need to know any more than I've put on the slides. It's just making it clear in the context of other things. So understanding why integration matters, understanding how and why it's linked to reverse transcription. So now we'll get into the real kind of nitty-gritty of retroviral genome expression and replication. So again, we're talking about retroviruses, we're talking about class 6 on the Baltimore classification system. We did touch on genome expression for retroviruses previously, but we didn't go into sufficient detail really, and we didn't touch on retroviral genome replication at all. So it does make sense to consider both of these together today in quite a bit more detail. So just a recap of where we're up to so far. We start off with reverse transcription. So we start off with a single strand of RNA being used to produce a double strand of DNA. And this is mediated by an enzyme called reverse transcriptase, which is an RNA-dependent DNA polymerase using an RNA template to produce DNA. Then we have a process called integration, and integration is the insertion of this double strand of DNA into the host cell genome. This process is mediated by an enzyme called integrase, and this enzyme is not a polymerase. It's also key to understand that integrase and reverse transcriptase are packaged inside the HIV particle. So as HIV enters into the cell, it contains reverse transcriptase and it contains integrase. It's also important to note at this point, and we'll talk about this in later lectures a bit more, but this specific step, reverse transcriptase, and to a lesser extent integrase, is an extremely effective and extremely attractive therapeutic target. So when we're thinking about therapeutic targets, we're thinking about processes that we can target with drugs, really. We're talking about processes that we can try to disrupt, and in doing so can prevent infection. When we're disrupting the specific processes, we're presumably disrupting infection. That's the idea, because we're preventing the virus from replicating successfully. The issue with targeting specific processes with viruses is that viruses often co-opt host machinery, so they conduct the same processes as the host cell, because that's the whole point of viruses, if you like. That's the whole driving force of their existence. They have evolved parasitic life cycles. They've evolved to co-opt host machinery. So it's quite difficult to target viruses therapeutically using drugs without targeting host cell processes as well. If we use translation, for example, we know viruses use host cell translation machinery. We've discussed it at length. If we therefore used drugs that inhibited viral translation, we would also be inhibiting host cell translation, and that's a huge problem, because that's what we call toxicity. It's a side effect. That's what it's usually experienced as. And side effects can be, sometimes we're talking about nausea, and like kind of mild things, but at the other side of the spectrum, we're potentially talking about drugs that are extremely toxic, and obviously that's a huge problem. We don't want drugs that are having side effects, even mild side effects. They're problematic because people become discouraged from taking drugs. People are disinclined from taking drugs that have side effects, even if, as I said, we're talking about mild effects. We're talking about nausea and things like that. So ideally we want drugs that don't have side effects. We want drugs that have extremely low toxicity. And one of the ways we can develop these drugs is to specifically target viral processes that are unique to viruses, so that we don't see in healthy cells. So we're not targeting cellular processes, we're targeting specifically viral processes. Now, reverse transcription, and to a lesser extent integration, is a very attractive target because it's exclusive to viruses. We've said we never have the process of reverse transcription happening in a healthy cell, in an uninfected cell. There's no cellular RNA-dependent DNA polymerases, that they don't exist. So the process of reverse transcription is never happening in a healthy cell. It only happens in viral infection, specifically infection of viruses that engage, infection by viruses that engage in the process of reverse transcription. So a large number of antiretroviral drugs, so drugs that target HIV specifically, target the process of reverse transcription. And when we're talking about therapeutics, we'll talk about this in detail. There really are quite a large number of drugs that specifically target this enzyme, that specifically target reverse transcriptase. There's also a number of drugs that target integration, so they target the viral integrase for the same reason. And again, that reason is because these are specific viral enzymes mediating specific viral processes. So if we can target those processes and enzymes specifically, we can hopefully limit the side effects of a drug. So next up, we're thinking about genome expression and replication. So the events we've talked about so far, they're really precursors to genome expression and replication. These are events that happen before genome expression and replication can occur. What we're looking at in the bottom half of the diagram here is genome expression and replication, really summarized. The enzyme is a DNA-dependent RNA polymerase, which makes sense, right? We're talking about after integration. So at this stage, the viral genome is DNA, because it's been a DNA copy of the genome has been produced from the RNA template, and this DNA has then been integrated into the host cell genome. And this is the site from which genome expression and replication occurs. So it's not the original RNA genome that is directly expressed and that is directly replicated. The process occurs via the DNA version of the viral genome that is integrated into the host cell genome. And again, this is what I'm trying to explain in the bottom half of the diagram here. Again, that also explains why the enzyme that's mediating genome expression and genome replication is a DNA-dependent RNA polymerase. So it's using the DNA version of the viral genome that's integrated into the host cell genome to produce RNA transcripts. So again, a DNA-dependent RNA polymerase. And it can seem a little bit confusing at first how HIV, even though it's a single-stranded RNA virus, is using a DNA-dependent RNA polymerase to express and replicate its genome. But again, the reason is because this genome expression and replication is happening after reverse transcription, and it's happening after integration. So at this point, the viral genome is in a double-stranded DNA form. So it makes sense that a DNA-dependent RNA polymerase is used to express and produce copies of the viral genome for replication. So next we want to differentiate between genome expression and genome replication, and interestingly, perhaps expectedly, for HIV, for these retroviruses, we see the same we see for the other RNA viruses. Often, not all the other RNA viruses, sorry, but for a lot of the other RNA viruses, we see a switch from genome expression to genome replication. That is practically a switch from the production of subgenomic fragments to the production of full-length genome copies, and that's what's represented at the bottom of the diagram here. On the right, we have these subgenomic fragments being produced, and this is genome expression. On the left, we have a full-length copy of the viral genome being produced, and this is genome replication. And if we remember, the reason why this has to occur is because these subgenomic fragments produced for expression, they can be translated just fine, no problem. So these are absolutely sufficient for genome expression, absolutely sufficient for protein production. However, for genome replication, it's necessary to produce full-length copies of the viral genome. So this requires a switch to the production of full-length copies. The production of the subgenomic fragments just isn't sufficient. And again, on the right, this is genome expression, and on the left, genome replication. So what mediates this switch? How does this switch occur? Just an extra bit of terminology that I need to explain. You'll see this term provirus used sometimes, and provirus specifically refers to the viral genome integrated into the host cell genome. So pro is, before, really, and we use this term because the viral genome is integrated into the host cell as a step before the production of new virus particles. So as a step before the production of replicants of the genome and a step before the expression of the genome. So we use the term provirus. So we want to look at genome expression in a bit more detail. As with many other single-stranded RNA viruses, HIV is utilizing subgenomic fragments for genome expression. Again, this is absolutely fine because some subgenomic fragments can be translated into protein. It also allows the virus a bit more control over the expression of its own genome, in that it means different genes can be expressed at different levels, so we get differential expression. The mechanism by which these subgenomic fragments are formed is essentially splicing. And splicing is normally a cellular process. So again, this is a virus utilizing a cellular process, a virus manipulating host cell machinery for its own benefit, for its own purpose. So in HIV genome expression, we initially have full-length transcripts produced, always. So the HIV genome is in a DNA form integrated into the host cell genome as a provirus. This provirus is expressed and full-length RNA transcripts are produced. These full-length RNA transcripts are then spliced, and they are spliced by host cell machinery, by the spliceosomes, and the splicing results in subgenomic fragments, so results in multiple fragments that are, of course, of shorter length than the entire transcript. These subgenomic fragments are then translated, again using host cell machinery, which is the case for all viruses. This translation results in the production of various viral proteins, one of which is REV. And this is probably starting to feel a little bit familiar now, because it really is quite similar to the strategies utilized by the other RNA viruses that have a switch from genome expression to genome replication. You'll remember a lot of RNA viruses that have this switch, they have evolved a specific switch from genome expression to genome replication that is triggered by the accumulation of particular viral proteins. And that's the case for HIV as well. So as the genome is being expressed, this specific viral protein called REV builds up. So the levels of REV build up within the cell, and there's a consequence for that, which we're going to see now. Once the REV proteins build up, they ultimately bind to what's called an RRE, which stands for the REV response element. And the REV response element is a sequence on the full-length genome transcript. So the full-length RNA transcript that is produced during genome expression of HIV. It's important to note that REV binds to this RRE before splicing occurs, so it's binding again to the full-length transcript. Once REV is bound, it mediates the export of the full-length transcript out of the nucleus. Now, splicing is something that happens in the nucleus. So by mediating the transport of full-length transcripts out of the nucleus, REV is essentially preventing these transcripts from being spliced. So what happens is REV binds to the RRE on the full-length transcript. The full-length transcript is then exported into the cytoplasm before it can be spliced, and this full-length transcript can then be incorporated into new virus particles because it is a full-length copy of the original HIV genome. If we remember, the original HIV genome is single-stranded, positive sense RNA, and that's exactly what this product is at the end here. And again, remember the copy of a genome, so the product of genome replication has to be the same as the original genome. And that's exactly what we're seeing here. When REV binds to the REV response element, it mediates the transport of a full-length genome copy out of the nucleus into the cytoplasm. And this copy can serve as the new genome for new HIV particles that are being assembled within the cytoplasm. Unfortunately, it gets just a little bit more complicated because the situation isn't quite that black and white. And what I mean by that is these full-length transcripts, though they are copies of the genome and though they can be packaged into new virus particles, they can also sometimes be translated, which makes sense. I mean, there's nothing to stop them being translated. They're RNA and they're positive sense. So even though they are full-length copies of the original viral genome, so they can serve as new genomes for the new virus particles being made, they can also be translated. Next up, there's an additional step which makes it a little bit more complicated still, but it should make sense. It's a middle ground between genome expression and genome replication in a way. And what I mean by that is as REV levels build up within the cell and REV starts binding to the REV response element, we can see partially spliced transcripts being exported from the nucleus. So when we were talking about genome expression, we were talking about how the transcripts that are produced are spliced in the nucleus. They're spliced into subgenomic fragments, and these subgenomic fragments are then translated into protein. For genome replication, we contrasted that and said Rev has built up in the cell at this point, and Rev is therefore able to bind to RRE. And when it's bound to RRE, the full-length transcripts are exported into the cytoplasm before they are spliced, and that's why these are full-length transcripts in the cytoplasm. There's a middle ground, so Rev can bind to partially spliced transcripts. So these are transcripts that aren't fully spliced, so they're not completely cut into subgenomic fragments, but they are partially spliced. If Rev is binding to these partially spliced transcripts, these partially spliced transcripts will be exported into the cytoplasm. So we have this additional step in between the switch from genome expression to genome replication. When these partially spliced transcripts are translated into protein in the cytoplasm, we see what we call mid-stage genes are translated. So mid-stage proteins are produced. So the proteins produced by these completely spliced genomic fragments are different to the proteins that are produced by these partially spliced fragments. So again, this is really a strategy utilized by the virus to control its own gene expression. So it's going from early stage genes to mid stage genes, and then when the full length transcripts are expressed, at that point late stage genes are produced. And here this information is just being reiterated really. It's this idea of partially spliced transcripts being transported out of the nucleus where they can be translated in the cytoplasm. And this is occurring as Rev levels are building up, but they're not high enough to mediate the transport of full-length transcripts. So you can kind of think of it as there's not enough Rev binding to transport the full-length transcripts into the cytoplasm before they're spliced. So Rev levels need to be high enough to mediate the transport of transcripts out of the nucleus fast enough that there's no splicing occurring. If there's less Rev, then there's some opportunity for splicing to occur before transport out of the nucleus can happen. And again, just this idea that we have early genes, the early genes that are expressed through these subgenomic fragments. Then we have these mid genes that are expressed through the partially spliced transcripts. Then we have the late genes that are expressed through these full genome length transcripts. So the virus is therefore able to maintain some kind of control. It's able to express different genes during the different stages of infection. So it's able to express the early genes when it's establishing infection. So immediately after the DNA version of the genome has been integrated into the host cell genome. Then it's able to express the mid-stage genes, and these mid-stage genes are involved in maintaining infection. So these are playing a different role in infection. And then the late-stage genes are expressed last from the full transcripts, and these late-stage genes are typically involved in preparing for the next round of replication. So it's clear how the virus is benefiting from the ability to express these genes differentially, early genes, mid genes, and late genes. We can kind of think, but it's the same genome, so how are these genes different? The answer is because there's what we call different open reading frames. So in any single strand of nucleic acid, you can have the potential to encode 3 different genes kind of over the top of each other, because we can have different reading frames, because remember we're working in threes, so codons are in triplets. So if we have a sequence of nucleic acid here, we can just straight split it up into threes, and then we can kind of work out genes that way, because so many codons make up a gene. However, if we start counting one base in, so we slip into a different reading frame, then the triplets are completely different, right? So in the same region, we actually have a completely different amino acid sequence. And then remember, we're operating in triplets, so we can do the same thing again, and we can start on the third base. And then if we start on the third base, the first codon we're looking at is the third, fourth, and fifth bases. So this is a third open reading frame. And if we progress through the nucleic acid, we see that there are potentially 3 open reading frames that we can use, because there can't be a fourth open reading frame, because the fourth base is just starting the second codon of the first open reading frame. So there's a maximum of 3 open reading frames. And HIV utilizes all 3 of these. HIV and other retroviruses as well. They have 3 open reading frames that are utilized. So that's why the genome is able to encode more than one gene in the same space. Okay, so that's retroviral genome expression and replication. So that really applies to group 6. We're talking about the retroviruses there. Hopefully these intermediates are clear now, by the way. The reason why we have the RNA DNA intermediate, sorry, I'm not sure if I explained this, but if you look at the diagram of reverse transcription, so that very complicated diagram I showed, you'll see that halfway between the RNA transcript and the double-stranded DNA product, there is a duplex, which is half RNA and half DNA. And that's why we have a DNA-RNA duplex, represented here as DNA/RNA, as an intermediate of reverse transcription in this class 6 of the Baltimore classification system. And the endpoint here is, of course, double-stranded DNA, which can then be expressed, it can then be transcribed into RNA. So finally, we want to think about class 7. We don't want to do this at length. We just want to make sure we understand the key things here. The key family of viruses in group 7 are the Hepadnaviridae. So the Hepadnaviruses, and the family is called Hepadnaviridae. We're talking about some specific hepatitis viruses belonging to Hepadnaviridae. Not all hepatitis viruses belong to Hepadnaviridae, but some do, namely hepatitis B, which is obviously quite an important human virus. So that's what we're going to talk about now. If you remember the discussion we had about viral genome structures, either last week or the week before, I think it was last week, you'll remember that the Hepadnaviruses, so the members of the Hepadnaviridae family, have this circular genome. So it's a genome that is mostly double-stranded. It's composed of DNA, and there's a single piece of single-stranded DNA. So it's a double-stranded circle, but there's a little portion of the circle that is single-stranded. We therefore call it a gapped genome, because it's essentially a double-stranded circular genome, but one of the strands has a gap in it, which is why it appears single-stranded at a specific point. So we tend to refer to it as a gapped double-stranded DNA genome. The way this genome is processed in cells is this single-stranded region is essentially repaired. So the host cell repair mechanisms will repair this single-stranded region, and the end product of this repair process is a fully double-stranded circular genome. And that's what we see here, and we just see the repair process summarized. And this is the process of genome expression and replication. And again, we did cover this the other week. It's just I want to cover it now as well, so we have group 6 and group 7 together. So we can see there's a DNA-dependent RNA polymerase which uses the double-stranded DNA herpesvirus genome as a template. And again, this herpesvirus genome has been repaired, so it's a full double-stranded circular genome. A DNA-dependent RNA polymerase uses this double-stranded herpesvirus genome as a template because it's a DNA-dependent polymerase. It produces RNA because it's an RNA polymerase, so a DNA-dependent RNA polymerase, and the RNA that is produced is exported into the cytoplasm, where it can then either be translated into protein or it can be reverse transcribed into DNA. So it can be reverse transcribed back into a double-stranded genome that is gapped. So a double-stranded gapped genome. And we can see that these two options here essentially provide the option for genome expression. So we have translation into protein, or we have genome replication, because we have a circular genome that is double-stranded, however, that is also gapped. So this genome is essentially exactly the same, with the exception of possible mutations, to the original herpesvirus genome. So this isn't actually that complicated, really. It's fairly simple, particularly compared to the retroviral genome expression and replication. But we do need to be aware of this, so just make sure you understand what's going on here. Okay, so that's everything really in terms of genome expression and genome replication. We've kind of come to the end of this particular part of the module, to the end of this journey. We've gone through all the different groups. We've hopefully clarified how these different groups express their genomes and how they replicate their genomes. What I have now is just a collection of slides that essentially summarizes what we've talked about. I realise that wading through 3 lectures worth of slides can be a bit tiresome. You'll need to do it before the exam, that's inarguable, but it could also be useful to have a summarized version that you can just flick through. So here we have just a few slides to summarize the ideas of genome expression and genome replication. So it's a diagrammatic representation of the processes, including the intermediates, because of course you do need to know what the intermediates are. It should be clear. I mean, I'm using the kind of systems we've used through the lectures so far in terms of the colors and the shapes to represent RNA and DNA and protein as well. So if you're looking at this now and it doesn't quite click, then that's a sign you should go back to the main content of the lectures and just have a think and make sure you understand these processes in detail. So this is genome expression we're looking at here. And then we have genome replication as well. So yeah, hopefully this is useful. Retroviruses and herpesviruses, so group 6 and group 7 aren't on this summary. Again, they're slightly different, they're a little bit more complex, so you'll have to consider those separately, but at least these slides are a summary of the principal groups that we've studied. Okay, so thanks very much. Again, I hope this is useful. I'll see you at the tutorial on Wednesday. Yeah, please come prepared with any questions that you have. Thank you very much.